Okay, so let's start. I always like to start the reviews with just to see if anybody has any questions. Is there anything that you for sure want me to go over? Is there anything that you're like, KJ, I have never understood genetic engineering experiments or gene transfer in prokaryotes or replication or whatever? Is there anything that you want me to go over?
Is it possible for you to go over a part of the test again, or no?
Yes, a little bit. I will not present when I do that, just because I don't like to have that on a recording. It feels a little bit, so, well, it's a long story, but I'm not going to go over it. But so yes, I will go over part of the test, but I won't present when I'm going.
Oh, because I have a specific question about.
Okay, tell me about your specific question. I'll see what we can figure out.
It's the one with, I think it has like the TRP operon and like the YPF, and then there's like different colors light.
And I did use that question again. But I've changed it slightly, so it's not exactly the same. I'll tell you what I changed is which operon we're using. So I'm not using the trip operon this time. I'm going to use a different operon. But let me see if I can. Let me see. Here, I am going to present, and I'm going to present the trip operon anyway. Give me just a moment. Trip operon. And let's see if we can find some images of it. So let's try. Give me just a second to find my favorite trip operon image. That's Lac Operon. Ah, come on. Okay, well, we'll just go with this one. I'm hoping this is a good image. I don't like this image. Sorry. Okay, I'm going to go with this image there. There we go. We're going to go with this image. Okay, so just really quickly. So with that one, the trip operon, with that particular question on the test, what they said was that they had changed this first gene, the TRPE gene. They, instead of having that gene, they inserted a gene for the YPF, the yellow fluorescent, YFP, the yellow fluorescent protein. And so if RNA polymerase was able to read across and read that gene and read the other genes, then it would trigger the yellow fluorescent protein under ultraviolet light. You can't see it under white light. So it won't look like anything until you put it under ultraviolet light, and then all of a sudden it glows. So the part that a couple things that you needed to understand was, when is the trip operon on and when is it off? And then in the story, it said that normally the gene is on. Normally the operon is activated because tryptophan is a hard amino acid to get in the diet. So as long as it's on, as long as there's tryptophan, I'm going to say that differently, as long as there's no tryptophan in the environment, if there's no tryptophan in the environment, then this repressor is gone. And the RNA polymerase is going to read across, and it's going to help the cell build tryptophan. But if we do put tryptophan in the environment, then the repressor clicks on, and RNA polymerase doesn't read it. It bumps into it, beep, beep, let me through. But it doesn't read these genes because there's no point in making tryptophan if there's tryptophan in the environment. So in the experiment, we made a genetically engineered plasmid that had two things. It had the trip operon with this changed to the yellow fluorescent protein, and it had a gene for resistance to antibiotics. So in that experiment, what you do is you mix the plasmid with some bacteria in the same environment. And some of the bacteria suck up the plasmid and some of them do not. The ones that don't suck up the plasmid, you can kill them because you don't want to study them. You only want to study the ones that sucked up the plasmid. You can kill those by adding the antibiotic. So if I have my Petri dish full of bacteria, some of them have sucked up the plasmid. That means that the ones that sucked up the plasmid, they're antibiotic resistant. So they can survive if I give them an antibiotic. So I'm going to pour an antibiotic on my Petri dish, and now I've killed all the ones that did not absorb the plasmid. That's the purpose of the antibiotic, is to kill the ones that didn't do what you needed them to do. Because you can't look at a bacteria and know if it's got the gene or not under normal circumstances. With the yellow
circumstance, was penicillin the antibiotic?
In your example, yes, it was. On the test example, it was penicillin. That's correct. So then some of you may or may not remember in that circumstance, you were given 4 different scenarios. One where there was penicillin and tryptophan in the environment, one where there was no penicillin and no tryptophan in the environment, and then one where there was penicillin and no tryptophan, or no penicillin and tryptophan. And so you needed to look at the different colors to see what was happening. You actually should have been able to do it in your head. You shouldn't even need the data table, but the data table helps to look at, to help organize your thoughts when you're in a hurry. And so the one that had, anytime that there was no tryptophan, look at, there's tryptophan here. If there's no tryptophan, then the RNA polymerase is going to move across and there's no repressor. If there's no tryptophan, there's no repressor. The RNA polymerase is going to move across, and those bacteria that have this plasmid, they're going to look yellow under ultraviolet light because the RNA polymerase will have gone across. If there is, let me make sure I say that right. If there is tryptophan present, there's the tryptophan, the RNA polymerase is not going to move. It is not going to read the yellow fluorescent protein gene, and those bacteria will not glow, not because they didn't get the plasmid, but because tryptophan is present and it stopped, it repressed that operon. So the only way to get them to glow is to make sure there's no tryptophan in the environment.
And was the question just how do they glow, or like what gets them to glow? Because I don't remember.
So there were 3 questions. The first question was, and it depended what test you got. What's the independent variable? What's the dependent variable? So what they purposely changed each time was the penicillin and the tryptophan. That's what they were purposely switching each time. And the dependent variable was whether it glowed, what color it was. That's what they were measuring, whether it changed color. And then it asked you about the purpose of the antibiotic. You don't have to have the antibiotic in there, but the antibiotic, if you put the antibiotic in there, it kills the bacteria that never sucked up the plasmid in the first place. So it gets them out of your way when you're trying to study the consequences of having this plasmid. And then the last question was, why are some of them glowing? And the reason that some of them are glowing is because there was no tryptophan present. The ones that did have tryptophan present, the RNA polymerase could not get through and it couldn't read the glowing gene. So none of them glowed. But the ones where there was no tryptophan, then the RNA polymerase could go through, it could read the glowing gene, and those guys glowed. It had nothing to do with the antibiotic. The antibiotic is optional. It just gets some of them out of the way. What decides whether they glow or not is whether there was tryptophan present. Now, when you take this, when you have this question again, it's going to be a different operon. So there's going to be a different chemical that's present or not present, and that's going to be the deciding whether it glows or not. There's also going to be a different antibiotic, which again, the antibiotic is to kill the ones that didn't absorb the plasmid, but it shouldn't have an impact on ultimately on the whether anything glows or not. Okay, other questions or topics that you want me to go over? Liz.
So what's the difference between transcription and translation?
I hate those words. I hate them. Transcription, transcription, I was trying to see if I could show you. Transcription is making RNA. Here we go, perfect. So when one side of the DNA gets copied and we make RNA, that's called transcription. And then the RNA gets read by the ribosomes, and they bring the trucks, the tRNAs over, and they drop off their amino acids. That's translation. So building the protein, that's translation. The only way that I remember it is this dumb letter R right there, transcription and RNA. I know that's terrible. I've been teaching this for 20 years. That is all I can do to remember. Does anybody have a better way? Like I know the definition of transcription and translation, but that does not help me remember which one is making RNA and which one is making the protein. Like, I need a better trick than that, and I don't have a better trick. So that's the answer. Do you have any questions about that, Liz?
No, that makes sense. I just didn't know how to determine which one was different from each other, because I understand how they work. I just didn't know which one was which.
Those two words, like I hate those two words. I prefer, instead of using the word translation, I prefer to say protein synthesis. To synthesize means to build something, so you're building a protein. That makes sense to me. Transcription and translation, they sound too much the same. It's hard for me to keep track of them in my head. All right, another question. I'm happy to start from the beginning, but if you guys know you have a question, let's hear them. Adam, Will, Maya, you guys, anything?
Could we talk about the one FRQ question about the sulfur and the nitrogen, I think?
The sulfur, oh, the experiment. And yes, I'm including that question again. I'm just changing it a little bit. So that was, give me a second. That was the Missense nope, that wasn't. That was, give me a second. I'm suddenly forgetting who did the experiment. Why am I forgetting who did that experiment, you guys?
Oh, I don't have the answer, but was this question the one on the FRQ?
Yes.
Okay.
Protein versus nitrogen, no, versus DNA. I can't believe I'm forgetting who did this experiment, you guys. I'm really sorry. This one, there we go. Hershey Chase. Hershey Chase. Okay, so the Hershey Chase experiment, that's Hershey and Chase right there. So the idea here, remember, is that they used sulfur one time. And the reason that they used sulfur is that there's no sulfur in DNA, but there is sulfur in some amino acids, cysteine, methionine, and I can't remember, have sulfur in them. So if they use radioactive sulfur, then the proteins are going to be radioactive. So as they're trying to figure out what was the genetic material, what was the hereditary material that these viruses were passing to the bacteria, The first time they used sulfur and they spun it down in a centrifuge, they blended it and then spun it down, and they saw that the contents of the bacteria had no radioactivity. So that meant it was not protein that got injected into the bacteria. And then they did the experiment again, but they did it with radioactive phosphorus. Phosphorus is in DNA, but not in proteins. It's in that sugar phosphate backbone of the DNA. So this time when they did the experiment and they blended and then they spun it, the pellet, which is where the bacteria were, was radioactive. And that told us that the DNA is what the viruses had been injecting. That was the hereditary information. It was DNA. So on the FRQ, I said, that was the experiment. Why didn't they use nitrogen? And the answer was because nitrogen is in both of these. There are nitrogen bases in DNA, A, T, C, G, and U. And there's nitrogen in amino acids. The amine group is an NH2 or NH3 group. So nitrogen is in both of them. So they would have been radioactive. The pellet would have been radioactive both times. So it's good that you asked me that, Aiden, because I'm going to ask you almost the identical question on the retake. Do you feel like you understand it, you guys? Any questions about it? What else? I'm happy to review anything. You know, we can do a real review, but just as long as you have questions, I'm always happy to answer the questions. Okay, why don't we just, gosh, I don't have my Prezzies pulled up at the moment. I got to think the best review for you guys. Let's go ahead and walk through the Prezzies. I'm just going to have to pull them up from our review page. So give me a second to remember where I put our review page. So modules. I did make a review page, but I just don't think it's here specifically. Oh, there it is, study resources. That's what we're looking for. Okay, so let's go ahead and click on the first Prezi. Well, it's not necessarily the first one. I think it was. This is the one that just reminded us about sort of the basics: replication, transcription, translation, central dogma. That was a question that lots of people got wrong. Just remember that the reason that we can do a genetic engineering experiment, the reason that we can put a human gene into a bacteria's DNA and it works is because all living things on this planet use the same genetic code. And so A, T, C, G, and U, that's what the bacteria use. And so do we. And so we can insert our DNA. The one step that I never really taught you and nobody asked me about, but I'm just throwing it out there. When we do a genetic engineering experiment, this is a great review. Human DNA has introns and exons. Bacterial DNA generally doesn't. So if I just take the raw DNA for insulin, or human growth hormone, or some other human protein, and I put it right into the bacteria's DNA, it doesn't actually work because the bacteria don't know what to do with introns. So we actually have to cut out the introns, splice them together, and then we get to put that gene of interest into the bacteria. That was beyond the scope of this class, but it does help you remember that that's a key difference between eukaryotes and prokaryotes, is that eukaryotes have those introns, and prokaryotes generally don't have those introns. All right, so central dogma is just the idea that all living things on this planet transfer information from DNA to RNA to protein. What is the, somebody shouted out, the only exception to that we talked about in this course? What's the only exception? And even then it's very similar, but it's a little bit different. What's the only exception? Anybody, can you shout it out?
Retroviruses.
Retroviruses. So retroviruses like HIV actually inside, so that experiment that we were just looking at, this one, instead of DNA that they're injecting RNA. So they had DNA in this case. The experiment still would have turned out the same if it was DNA or RNA in that case, because they have phosphorus in both DNA and RNA. But retroviruses are the only ones that use, where did my tab go? There we go. Retroviruses are the only ones that start with RNA. And then just reminding you quickly, they have reverse transcriptase, which takes their RNA and converts it into DNA, but it screws it up royally, all kinds of mutations. And that's why we have such a hard time making vaccines for retroviruses, because everybody has a different variety of it within their bodies. And so the secret to those kinds of vaccines is trying to figure out what is it that makes HIV the same in all the patients and what is actually changing. And so our vaccine has to focus on the part that's the same. How do we know it's HIV if it's always changing? There's got to be some things that are the same, and that's what the vaccine kind of has to focus on. All right. So replication, you guys, remember replication. I'll just zip through it quick. Replication is just the idea that we have to copy the DNA so that it can divide. So we have to go through replication in order to go through mitosis. And replication is the #1 source of mutations. Our cells are super good at copying the DNA, and we have proofreaders, which it talks about down in this corner. We have proofreaders so that in the end, we only have like 1 mutation every 3 cells, every 3 cell divisions. However, in an organism that's made of 100 trillion cells, by the time you get to be 80, you have a lot of mutations that have accrued. And some of us are lucky, and those mutations are silent or don't end up being important for one reason or another. Some of us are less lucky, and those mutations end up being in places that control cell division. And so those end up leading to cancers. But it also is part of what makes us look like old people and even act like old people. The more mutations we get, the less efficiently our bodies are working, the more mistakes our bodies are making, and what have you. Gravity also has something to do. Gravity is why our eyes go bad with time. Gravity is why our skin sags, but there's more to it than just gravity. All right, so with replication, trying to think if there's anything. Oh yeah, we need to know this experiment. So this is sometimes described, the Missense stall is sometimes described as the most beautiful experiment in biology. You did an entire assignment on this one where you had to show that semi-conservative was the correct form of replication. And semi-conservative means that one of the strands is old and one of the strands that gets copied is new. And so you need to understand sort of the principles behind this experiment. Hint, hint, it is on the retake. This experiment is on the retake. So I want you to remember how they did it. They started out with heavy nitrogen, and that's where they had the initial DNA. So the parental DNA was heavy. So when you spin it in a centrifuge, this DNA sinks low because it's got an isotope that's heavier. So take a look at this diagram. So then they allowed it to reproduce over different generations. And as the DNA is being replicated, the first time there's just one amount, and it's an average between 14 and 15. So that implied if it's an average between 14 and 15, that told us that it could not be the conservative model because that would produce a light band and a heavy band, period. So it could have either been the semi-conservative because this has heavy and light combined and this has heavy and light combined, or it could have been dispersive, which has a mixture in both of the copies. So these two we couldn't exclude from each other. So then they let the experiment go another generation. They actually did it several generations, but by the second generation, we knew the answer. By the second generation, we still had some that were in between the light nitrogen and the heavy nitrogen. So they were a combo, but we had some that were only light and had no heavy nitrogen in them at all. And so that made sense if you had semi-conservative. So here you had two, they split, and now this light blue one gets copied and we make another light blue one. So it's totally light. And same with this one, the light blue one gets copied with another light blue one. And so it's totally light. And then there's still the old one that needs to get copied with a light one, and this old one that needs to get copied with a light one. So you have one that's medium and one that's light. And that's the way it turned out, and that's how they proved it was semi-conservative replication. Any questions about that experiment, you guys? Really pretty, like when you think about who thought of these things, I have to say that I'm super impressed. Like I wish that I could say, oh, I would have thought of that. And it's so simple and beautifully and just brilliantly done. And so kind of the essence of good science. Okay, so replication, things that you need to make sure you know about replication. You need to know DNA polymerase, helicase, primase, topoisomerase, and ligase. That one's not mentioned right here. So DNA polymerase, write it down if you haven't already. DNA polymerase reads, there it is, or that, I guess they don't show DNA polymerase yet, but it reads 3' to 5'. But it builds 5' to 3'. And I think that that's more obvious in a different diagram. Is it this diagram? Sort of. So here's your DNA polymerase. That's the peach one. So this was the over here, this was the 3' corner. And now it's 5'. The light blue is 5', and they're going this direction. And if you go all the way, you see that the 3' is over here. So, and then on the dark blue strand, the 5' is on the top. So it reads the DNA A, T, T, A, C, C, G, G, T, T, A from starting in the 3' and going to the 5' direction. But as it's reading A, T, T, it's adding T, A, starting on the 3' and working toward 5', like it's building, sorry, I said that backwards, starting 5' and building 3'. So the newly replicated side is 5' to 3'. All right, and then just a quick review, topoisomerase untwists, helicase unzips, so breaks the hydrogen bonds. Remember that the bonds in the backbone are those really strong covalent phosphodiester bonds. So those bonds don't usually break. But the hydrogen bonds, super weak, and we can separate the two sides. Primase, sorry, trying to not move around so much. Primase has to set down the DNA RNA primer. If I'm not mistaken, I don't remember this for sure, but I think that the reason the primer gets put down is so that the DNA doesn't close back up on itself again. I think that that's why the primer is there, is just so that the DNA that's been separated doesn't do this again. So the primer gets in the way, and then the DNA polymerase is going to hop on and head toward the replication fork on the leading strand. And on the lagging strand, it's going to hop on. So if primase was here, I think I can show you that on this diagram over here. Here's the primase, so it's moving this way, and then DNA polymerase would hop on right here, and it would move this direction. As you can see it moving that direction in this diagram right there. And then the only other one we haven't talked about is ligase. Because ligase is necessary because the Okazaki fragments, if you notice the little gaps, they haven't been covalently bonded to each other, so they can fall apart because those hydrogen bonds are the only thing that's holding the Okazaki fragment there, and that's not very sturdy. And so ligase has to come through and create a covalent bond between the Okazaki fragments so that they don't literally fall off the strand. Okay, so that's replication. Any questions about replication? Okay, moving on, let's talk transcription, RNA, and translation. Oops, sorry. Can I get it to work? Okay, transcription. Remember, this is the RNA polymerase. It only reads one side, not both sides of the DNA. And it starts at the promoter. That's where it sits. So sometimes I remember that. I always do things with alliteration. So I remember promoter polymerase. That helps me. And remember that promoters also have these Tata boxes. I didn't ask you about the Tata box, but I did see an AP, actual AP exam that talked about the Tata box. Such a dorky name. I know, I don't have anything to do with it. All right, so the RNA polymerase is reading one side. It's reading the template strand. Remember that there's a whole bunch of other names and it drives me crazy. If I could rule the world, you guys, I would have easier names to remember, but template I feel like is a good name. So the template strand is where the RNA polymerase is reading and writing the mRNA. So this is the mRNA that's forming. Trying to think if there's anything else you need to know on this page. There are transcription factors, and we'll show you those here. So those where they look like little purple grapes or something, the transcription factors help the RNA polymerase to bind to the promoter. So here's the promoter with the Tata box, and those transcription factors help. Then remember in, let's see, it's a few prezzies from now, the eukaryotic control, they then have the activators. I should pull that up so you can look at it and just remember. I'm going to pull them up side by side so you can remember that one. Eukaryotes. Because the whole idea of the activators just comes up, oops, another time. Any questions while I'm waiting for this one to go up? How are we doing time-wise? We're doing amazing. So under eukaryotes, how they control whether their genes are on or off. So that's controlling or regulating gene expression, deciding whether it's on or off. One of them is this pre-transcription. And that's the one where there are, so here's those transcription factors again. We just saw them on the other page. Here are the transcription factors. They make them sort of this light purple color, okay? So now here they are again, but these are just general transcription factors. In this circumstance, there are these enhancers, which are sections of DNA a long ways away. And if activators, they're molecules in the environment, click on, and a DNA bending protein is there to fold the DNA over. Now that brings the activators close to where the promoter is. This is the Tata box and the whole promoter. Now you bring all of them together to create your transcription, complex. Here are the activators. Here's the mediator proteins, and here are the general transcription factors. And those also help the RNA polymerase to bind and be more efficient. And so it makes sure that RNA polymerase is doing its job. It's not just sort of randomly bumping into it. It's forcing the RNA polymerase because for whatever reason, it's important that that protein get made, that gene be read. All right, so going back to where we were, transcription. Remember when you look at RNA, that RNA looks different. mRNA only is single-sided, but importantly, it has uracil, so it has U's instead of T's. And then the sugar in RNA is ribose instead of deoxyribose. So that's where it gets the name RNA instead of DNA. All right, many kinds of RNA. So remember, does it talk about the trucks here? No, it talks about those in the next one. So the other thing that I think is important in this section is just to remember, lots of people forgot about what happens to RNA in a eukaryote. So remember that eukaryotes have introns and exons. So the mRNA has all that crap in it. So it has to be spliced out, and prokaryotes don't do that. And then we also have to add the 5' cap, which allows it to escape the nucleus. And it adds that poly A tail, which also seems to help it escape the nucleus, but it also extends the lifespan of the mRNA. So the longer the tail is, it appears that slows down how quickly it gets eaten. Recycled is what I was trying to show you. It gets recycled a little bit more slowly if its poly A tail is longer. Okay, so then last section on this one is translation. Whoops, sorry. Oh my gosh, just work. There we go. Okay, so here it is again, Liz. Transcription is making the RNA, and translation is actually building the protein or the polypeptide. All right, so in the ribosome, remember that eukaryotic ribosomes are a little bit different than prokaryotic ribosomes. And one of the big differences, it's not just in their structure, but one of the big differences is that prokaryotes have what they call polyribosomes. Because they don't have a nucleus, the DNA can be transcribed into mRNA and then translated into a protein all at the same time. Whereas in a eukaryote, transcription is happening in the nucleus, and then the mRNA has to leave and it has to go to a ribosome in the cytoplasm. And so they are decoupled is the phrase. They're not coupled together. They're not simultaneous. So then on the ribosome, these are the trucks. Remember, those are the tRNAs, and they're carrying the amino acids. The sites are called A, P, and E. And I say it that direction instead of EPA, I say APA, because the RNA is entering from the A, P, and then the E site. So that's how it's going through. The ribosome is always shifting this way and the RNA is going that way. So the truck drops off its amino acid, and then this amino acid right here bonds, it's a peptide bond, makes a peptide bond to this amino acid here. And now this truck is free. It's not attached to anything. So it moves to the E site. It doesn't move. The ribosome moves and places it in the E site, and then it can exit. That's what the E stands for, is exit. So it can then leave. And then remember, they always recycle, so it'll go back and pick up whatever amino acid it had been carrying. It'll go pick that up so that it can be reused over and over and over again. Trying to think if that's really all of translation right there. There's a few other things I guess we should talk about. Probably don't need to know that part right there. Oh, the trucks, just making sure you recognize what they look like. So I've never seen trucks drawn this way before. That's kind of unique. But what's important is that an amino acid is on one end and there's an anti-codon. The opposite 3 letters are on the truck. So to help me remember that, I always go with alliteration. Anti-codon is on the tRNA. So this is another drawing of the tRNA, sort of a flattened version of it. Here's the anti-codon, and here's where the amino acid will attach. And then this is more of a 3-dimensional image of the tRNA. Here's the anti-codon, and here's where the amino acid lands. So you got, your brain just has to sort of wrap around the different artists are going to draw these diagrams in different ways. Okay, and then let's just look at what happens. I feel like it's pretty, like we've talked about it. So the ribosome clamps on, and it's always starts, this is important, it always starts with methionine. The codon, who can tell me what the start codon is? You don't have to memorize it, it just happens accidentally. Oh, it's right there, isn't it? I didn't notice it was right there. So the start codon is AUG. So the truck that pulls up, it has the opposite 3 letters. So it would be UAC. That's the truck that's pulling up there. It shows it right there. That's the truck that pulled up. And it's carrying methionine. And methionine is always the first one. And then it just keeps switching every 3 letters. Those are the frames. And it keeps switching every 3 letters until we get to the end, until we reach a stop codon. Remember, importantly, that the stop codons do not have an amino acid associated with them. So the start codon did, the stop codons do not. And then, oh, I wanted to look at this table, just make sure you feel comfortable. Here's AUG and it carries methionine. Here are the 3 stop codons that do not have any amino acid with them. Okay, and then right here, the last thing that's really different, I mentioned this one about how prokaryotes can do, there's the RNA, so this is Rna Polymerase, so this is transcription, that's what the red line is the mRNA, the blue line is the DNA. So this RNA polymerase is transcribing the gene, and immediately these polyribosomes are attaching on, and they are immediately building the purple lines, which are the polypeptide or the protein. So that's all happening simultaneously. Whereas in eukaryotes, that has to be separated from each other. The other thing I wanted you to know about the eukaryotes, they have endoplasmic reticulum, which prokaryotes don't have. And the rough ER has ribosomes embedded on it. Why? Because occasionally, many times, there is a signal at the top of the polypeptide that tells this protein called the SRP protein to grab it and transfer the whole thing to the rough endoplasmic reticulum, which then the ribosome finishes building the peptide inside of the ER, and then the ER can transfer it. It's the highway of the cell. It can transfer that polypeptide to some other location in the cell. Eukaryotes are like a universe, not quite, in comparison to a prokaryote. They're huge compared to a prokaryote, and so they need special organelles to help them transfer their proteins to the correct locations. Okay, I can't remember, do we do mutations? Yes, we do mutations in this one. So it's good to remember, it's good to remember your mutations. Silent mutations, when we're just talking about the actual A's, T's, C's, and G's. And remember, I think a lot of people got this mixed up. Mutation is only in DNA. I say that and now I think a little bit, do they ever say that an mRNA is mutated? I don't think so. But remember that mRNA, even if the mRNA has a mutation, it has a limited lifespan. So it's only going to be mutated for a matter of, I don't know the lifespan of an mRNA, but a minute? I don't know. I don't know how long an mRNA lasts. Whereas a mutation that happens in the DNA, that's lifelong. That's as soon as that mutation happens and it doesn't get noticed, it's there. And so that lasts the life of the organism. So anyway, it's a DNA thing that's happening when we talk about mutations. And so let's take a look maybe a little bit more closely. So here's the DNA. And here's the DNA again, but they've changed one letter. So they substituted, pay close attention to whether the word is substitute or insert or delete. just substituting, not as big of a deal most of the time. So just replacing, this one was supposed to be a G and now it's an A. So across from an A goes a T. So now the two strands have an A and a T instead of a G and a C. And now when we make the mRNA, there was supposed to be a C there, but instead the mRNA has a U in it now. So that means it's going to produce the wrong that is the wrong codon. But because of wobble, because the third letter isn't that important, it didn't matter. And this one was silent. We still got glycine, whether it was GGC or GGU, it was fine. Then then there are missense mutations, where one letter changes and it actually does change the amino acid. And how bad that is depends. We know sickle cell anemia and cystic fibrosis are easy examples. One letter change, one amino acid change definitely has a genetic disorder. Just one letter creates a genetic disorder. Whereas lots of times it's silent and it doesn't affect anything. And then with nonsense, it just means that the mutation leads to a premature stop signal. And so the whole protein doesn't get to be made because we reach a stop signal early. And then finally, the frameshift mutations, that's specifically caused by an insertion or a deletion, not by a substitution. So if we insert or delete 1, 2 letters, we screw up the frames. If we insert or delete 3 letters, then the frames reset. So any multiple of 3 is less damaging than something that's not a multiple of 3. But in this case, extensive missense, we insert 1 letter right here, or I'm sorry, we deleted 1 letter right here, and that screwed up everything after it. All of the amino acids after that are screwed up, so that's extensive missense. Immediate nonsense means it produced a premature stop sign. And then limited effect is what they say, you don't hear about this one as often, but what they say if it came in a multiple of 3. So we're either going to miss an amino acid or we're going to add an amino acid, but it's not going to screw up all of the other amino acids. Now, does that mean it won't harm the organism? No, it doesn't mean that at all. It might not. It depends the chemistry of that amino acid that's either been added or that's been removed. Remember, their R groups are interacting with all of the other R groups, and the chemistry of that R group is what's going to determine whether it really messes with the folding of that protein. Are there proteins that are slightly misfolded that still work? And the answer is absolutely yes. So there are some folks that don't even know they have a genetic disorder because their proteins are mostly working right, but maybe a little bit messed up. And I could go into a lot of detail on that, but we don't have time, so we're not going to talk about that one. Okay, let's go back to this one because I think this is the hardest presentation, and then we'll see what else we need to talk about because I feel like we covered a lot. We talked about the Trip Operon already. Let's talk about the Lac Operon just to make sure you feel comfortable with it. Both of these are operons that we turn off. So that means they're under negative control or negative feedback. The cap activator, the name activator, is what makes it happen more and more and more. And so that's considered positive feedback or positive control in that circumstance. All right, so with the Lac Operon, it's normally shut off. Imagine a little E. coli bacteria in a bowl of water. There's no lactose, so there's no point in making the enzymes for digesting lactose, these ones right here. No point in making those if there's no lactose present. So we normally want, I shouldn't say we, because it's not in human beings. This is in a prokaryote. Human beings do not have operons. So there's a repressor that's got it shut off because there's not usually lactose present. However, if we scoop that little E. coli up and we put them in a bowl of milk, all of a sudden there's lactose present. It causes the repressor to fall off, the RNA polymerase can now read they're called structural genes. It can read the structural genes and make the enzymes for digesting lactose. Also, just an FYI, where does the repressor protein come from? Well, there's DNA upstream that codes for the repressor, so that there's always a repressor available. And then the repressor snaps back on when they run out of lactose. So once all the lactose has been digested, then the repressor jumps back on again. And then the last one, yeah, just the cap. Remember that the way that I try to describe cap is like if the RNA polymerase is like a car driving down the highway, cap is like a turbocharged engine, a really powerful engine that speeds it up. And then when is cap present? Cap is present if the cell is hungry. It doesn't need RNA polymerase to go super fast if it's already got plenty of food. It's fine if RNA polymerase does whatever. As long as it's full, it doesn't care. So, trying to make a point here, cap is present if there's not much glucose. Cap jumps on if there's not much glucose, if it's hungry. But if there's plenty of glucose, then cap isn't there. Now, I've said this before, I'm not wild about this diagram because it implies that the RNA polymerase doesn't attach at all. It does, it's just going to go slowly. So it will attach, but we say that that's moving at it says, I think it says here, little Lac mRNA synthesized. Do you notice how it says little? Like it implies that it's not doing it at all. It is. It's at a basal level, a low level. Okay, that's the prokaryotes. How are we doing time-wise? We're almost done here, you guys. For the eukaryotic control, we already talked about pre-transcription. What did I do? I downloaded it. I didn't mean to download it. Okay, DNA access. This is the one that has to do with epigenetics. Epigenetics is actually more complicated than this explains. So methylation and acetylation apparently can either increase or decrease whether the DNA is getting read. So it apparently varies. That's kind of new information. So here's what you need to understand. If the DNA, I don't care what caused it, if the DNA has twisted up more, then the RNA polymerase can't get to it. So we say it's off. If it's all twisted up, then we say that that gene has been shut off. So that's a way that we can regulate whether that gene is on or off or not. So heterochromatin is more tightly packed, so we say it is off. And then euchromatin is less tightly packed, sorry, trying to get there. And so we say it's been activated. If it's unwound, now the RNA polymerase can come back and read it. All right, so that's that one. We talked about pre-transcription. This is one you had a question very much like this on the test, and you'll have a similar one on the retake. So this is how cells differentiate, why you have a liver cell versus a lens cell, and they don't look alike, they don't produce the same proteins. That's because of these activators, partly because it's always a bigger answer, but different molecules are present in each cell, and those molecules are what trigger some of the proteins to be built and some of the proteins to not be built. So these activators are here, and it causes the albumin gene to be expressed. But different activators are available here, and so the albumin gene is not expressed, but the crystalline gene is expressed. And so it's the presence of activators. There are also where does it say it? There are also inhibitors that instead of activating, they can inhibit the particular protein from being built. So you can have activators or inhibitors. All right, we already talked about this one, the alternative splicing, about how you can cut out. So eukaryotic DNA has introns and exons, whereas prokaryotic DNA has only introns or exons. I was trying to remember to say that correctly. And then alternative splicing means that different exons can be put together in different ways. You can even leave exons out, and that allows the eukaryote to build different proteins. So if it doesn't want this particular protein, it's not going to put the exons together. It's going to put them together in a different way. So controlling that gene expression. Translation, we learned about microRNA. So teeny tiny little small RNAs and how they interfere. Be careful because interference could mean lots of things. This is the two ways that we learned they interfere. Number one, we learned that they can cut up mRNA that's not supposed to be going right now. So it can karate chop it so that those genes never get translated. It can also interfere by just hanging on. It gets in the way of the ribosome clamping on, so that prevents translation. So either it can cut up the DNA or it can prevent the ribosomes from clicking on, but either way, the proteins aren't getting made. And so that's a way of controlling the gene expression. And then the last one we talked about was just the ubiquitin proteins tagging old proteins that are not needed anymore, and then a proteasome recycling them. So recycling so that it can be rebuilt, some other protein can be built instead. So getting rid of proteins that they don't want in the cell right now. Okay, so it's 3:45, a little bit past 3:45. Can you think of anything that I did not talk about? I know there are more prezzies. Is there anything that we need to talk about? I feel like we talked, okay, Chargaff's rule, just reminding you that A and T are always the same and C and G are always the same in DNA, that the opposite, so if A is always across from T and C is always across from G, if you have 40% A, then you're gonna have 40% T. And then the remaining 20% would be the C's and the G's, 10 for C and 10 for G. That's in DNA. Biotechnology we didn't talk about. We talked about a genetic engineering experiment. We could PCR. I want you to just remember that PCR, the purpose of PCR, trying to jump here really quickly. The purpose of PCR is to copy DNA. So you could copy DNA if you had a gene of interest that you were studying as a geneticist, and you just got one copy of it. Now you can have it's millions, billions, I don't remember, hundreds of trillions, I don't know, of copies of it in a few hours. If you had DNA at a crime scene, you could copy it. If you had, trying to think, those are examples of what we use PCR is. Anytime that you would want to copy the DNA, that's what PCR is for. Genetic engineering experiment. Oh, and then electrophoresis, just reminding you of that one. Gel electrophoresis. Here we go. I'll try this one. So in this example, remember that these are the wells where the molecule gets, where we add the molecules. Usually if it's DNA, it's been cut up with a restriction enzyme so that there's pieces. DNA pieces are always negatively charged, but on our test, it didn't have to be negative. I mean, it could have been something other than DNA. If it's negative, it's gonna head toward the positive end. If it's positive, it's gonna head toward the negative end. Opposites attract. And then remember that the further it travels from the well, the bigger it is. It moves really slowly. But the ones that travel really far away, they're itty bitty. And they were able to scurry across the gel in the same amount of time that it took this one to go this far. This one got that far. And you can use that for DNA fingerprinting, for identifying genetic disorders, identifying a suspect and the DNA at a crime scene, for example. Those are things you can use that for. The only other thing that I can think that we didn't talk about, you guys, and I'm sure you're just like, oh my gosh, let us go, is horizontal gene transfer in bacteria. So just want to remind you of that quickly, and then we can be done. Bacteria can get new DNA in one of 4 ways. They can suck up DNA from the environment. That's called transformation. That's how we do a genetic engineering experiment, but that's a natural thing that all bacteria do. They can suck up DNA, and then if that DNA, for example, has the gene for antibiotic resistance, yay, that bacteria has been transformed, and now the antibiotic doesn't kill it anymore. Conjugation is kind of like sex. They have this structure that's called a pilus, and the DNA can be transferred from one to the other. It's usually a plasmid that gets transferred from one to the other. Transduction is when I always try to close up, and I can't close up on a presentation. This particular, do you see that there's blue DNA inside of that bacteriophage? It accidentally picked up some of the bacteria's DNA. And now when it went and infected a new bacteria, that new bacteria, it injected some of the DNA from the old bacteria. So that's a way, it's a rare way, but it works to get new DNA from another bacteria. And then the last way are these things that are called jumping genes. That's sort of the common person's word for it. They're really called transposons. So transposition is when a gene can move, literally, from the main part of the genome over to, so on humans, it can move from one chromosome to another. But in bacteria, it often moves from the genome to a plasmid. And then the plasmid can be exchanged through conjugation or can be sucked up during transformation. So there's ways that other bacteria can then get it. Okay, cuties, I'm sorry. Does anybody have any questions before we go?
Do you think
you can go, Eden.
I was just going to say, do you think you could go over what's on the retake?
Oh my gosh. I should never expect that we can do a review session in an hour because it's not enough time. Yes, but I'm going to stop presenting. So let me just have a minute to take a look. Anybody who wants to go, who's just like, oh my gosh, this has been a long enough day, go. You do not need to stay. But I will. Bye, Liz. Have a good one. But I will take a look at my test. So give me a second to find it. I'm not presenting, right? I'll be really mad at myself if I'm presenting. La, 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 la. Okay, Unit 6 retake. And it is all multiple choice. So the very first question is oh, no, it's not. I thought it was a gel electrophoresis. Sorry. The very first question is calculating the number of nitrogen bases. So it's very similar to the first question on the FRQ. So it said there were, I'll make up a number, 942 amino acids. You need to be able to tell me how many nitrogen bases are in that DNA to make that, lead to that amino acid. Any questions about that? Can I move on? You need to shout at me. I'm just going to zip through them. You need to shout at me. Yes.
So do you only add 3 for the stop codon?
That's correct. So the way that the College Board does that particular question, usually they make it worth 2 points. On the multiple choice, I could only make it worth 1 point. On the FRQ, it was 2. If you did 932 times 3, you got 1 point because you understood that the codons had 3 letters. And then if you added 3 more letters for the stop codon, then you got your 2nd point. However, it is also correct to add another 3 letters. And if you add another 3 letters, that shows that you understand that methionine, which is on the start codon, almost always gets removed. And so then your finished polypeptide is actually missing methionine. So you could do that one or not. So just do the times 3 plus 3 and you're fine. You'll have the right answer and you'll be good. Let's see. This next one is, I've got 2 different types of cells, like the liver cell and the lens cell question. And why do why do the liver cells produce albumin and the lens cells produce crystalline? So it has to do with the presence of activators. And so that's what you're kind of looking for. I have a Chargaff's rule question. So what the percent of DNA, percent of nitrogen bases should be. This one requires a little bit of critical thinking though, so it's not super obvious. You just have to kind of figure this one out, but it's Chargaff's. Here's a retrovirus question. And just remember that the big deal about retroviruses is that they go RNA, DNA, RNA, protein, and the enzyme that they use to go from RNA to DNA, which is backwards, is called reverse transcriptase, and it makes lots of mutations. Here's a question about the role of ligase. So we kind of talked about what that enzyme does. Here's a question about plasmids and what plasmids are and what their role is in bacteria. Here's a question, give me a second, about genetic engineering. So how you do a genetic engineering experiment, adding a gene of interest into a plasmid and then having the bacteria absorb it and be transformed. Give me a second. This one is, so this one gives you a little couple sentence story, and then you have to decide which biotechnology you should use to figure out the issue. So should you use PCR? Should you use gel electrophoresis? Should you use a genetic engineering experiment? Whatever. Some combination of them to solve this particular problem. So something about biotech. Here's the gel electrophoresis question. So are they negatively or positively charged, and how big are they, the fragments? Here's a question about, you guys had a question very similar about DNA replication, and it shows you a diagram with the RNA primer. So you need to know which direction the DNA polymerase is going to go. So you have to pay attention to which side, it could be upside down or right side up, which side is the leading strand and which side is the lagging strand, in order to know which direction the RNA or the DNA polymerase is going to go. Is it going to go toward the replication fork? Is it going to go away from the replication fork? It's going to go away if we're on the lagging strand. It's going to go toward if the RNA primer is on the leading strand. So you have to figure that out yourselves. Here's the question I told you about the Hershey Chase experiment. That's the name I couldn't remember. The Hershey Chase experiment about what they saw when they spun it in the test tube, the different bands and whether the bands were heavy or light. We did a whole packet on that. Here's a question on eukaryotic control. So eukaryotic gene regulation, those 5, we kind of just went over them, like epigenetics and pre-transcription and post whatever. Here's a question about the Lac Operon. Here's a question about a mutation, and I'm going to give you a, so be careful with this. I'm going to give you a strand of DNA. Careful. I'm going to give you a strand of DNA, and it says, what is the, give me a second to, what would be the effect of a mutation? You have to figure out where the start codon is, but it's written in DNA. Remember the start codon, okay, I think you all know the start codon is AUG, but that's RNA. That's the opposite of DNA. So when I give you the DNA, flip it around in your heads, what's the start codon in the DNA? Look for that, because that's the beginning of your gene, is where you see the start codon for the DNA. And it is not AUG. That's in the RNA. That's the opposite 3 letters. I'm not saying anything more than that. Here's a Frameshift question. Here's a Hershey Chase. So I told you about the the radioactive sulfur and the radioactive phosphorus with the protein in the DNA. So it's very, 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 very similar to the question you had on the previous test. Here's a question that just says, I don't know if I can say this one at all. Yeah, I'm not going to say anything about this one because if I said anything, it would totally give it away. Here's the question that Meredith asked about at the very beginning, which was the one that was the Trip Operon, and then it was combined with a genetic engineering experiment, and the yellow fluorescent protein. So that's this question. And oh, sorry, there's a story. So you have to read the scenario, and then there's 2 questions about that scenario. And that, my dears, is it. Any other preguntas before we go? I'm going to turn off the recording now because nobody wants to listen to such a long review session.